My name is Rebecca Desposito, and I'm a senior scientist here at Waters. Today I'm going to be showing you 3 of our RNA critical quality attribute, or CQA, analysis workflows. These workflows are data processing workflows, and they use Intact Mass, which is a Waters Connect application, and 2 of our Waters micro apps, mRNA Cleaver and mRNA Coverage Mapper. These workflows were designed with the intention to evaluate 3 main critical quality attributes of RNA-based biotherapeutics: sequence mapping, 5' cap characterization, and 3' poly A tail heterogeneity. With these workflows, we hope to ease the data analysis process of highly complex nucleic acid digests. The first CQA I'll be focusing on is sequence mapping. The workflow to evaluate sequence coverage after endoribonuclease digest utilizes all 3 programs I mentioned earlier: Intact Mass, mRNA Cleaver, and mRNA Coverage Mapper. The dataset I'll be using is a 100-mer single guide RNA, which was digested using RNase T1. Here is a high-level view of our workflow for evaluating nucleic acid CQAs. Typically, we start with the nucleic acid digest using an endoribonuclease such as RNase T1. Then we submit our digested sample to ion pairing reversed phase LCMS analysis on the BioAccord LCMS system with max-peak high-performance surface. Our data is acquired using the Waters Connect informatics platform, and once collected, we can move on to our fragment mapping data processing workflow. The data shown here is of a 100 nucleotide long single guide RNA that has 3 post-transcriptional modifications. The black trace is of an RNase T1 digested sgRNA, while the red trace is of the intact sgRNA. The TIC traces are overlaid to demonstrate that the single guide RNA has been completely digested. The digestion fragments are smaller and elute well before the intact peak would on the same gradient. The gradient used is a shorter gradient than one would use with an mRNA sample, which are typically longer in length. Next, I'll show you how this data is processed using our fragment mapping data analysis workflow. First up is mRNA Cleaver. mRNA Cleaver is an in silico digest calculator that will generate our library of digestion fragments. After adding in our sequence and adjusting the settings to reflect our experimental parameters, we can hit calculate, and mRNA Cleaver will generate a list of theoretical digestion fragments. For this workflow, you will want to uncheck filter duplicate sequences. Having this checked will affect how the data is visualized in the last step. Then we will process the data in Intact Mass. First, we can select our method and our injection. Then hitting next, we can then add in our expected masses by copying in the molecule IDs and expected masses from the mRNA Cleaver generated library. Once we've copied that information in, we can then hit process. And here is what the results look like. The identity and purity tests will almost always fail, as the theoretical list of digestion fragments will include some fragments that can't or won't be identified. You can ignore the big red X. Clicking into the Results tab, the page will load, and then we'll be able to visualize our results. We'll have our TIC, TUV, and results table, and underneath the results table will be the deconvoluted masses that are found under every peak. Once the page is loaded, you can click on the download button located here. This will download a CSV of all of the tables: results, TIC, TUV, 260, and MS. The table that we need is the results table. That will be used in our next step when we visualize our data and calculate our coverage percentage. Finally, you can visualize your data with the Waters MicroApp mRNA Coverage Viewer. First, input your sequence. You'll see that the sequence has automatically been added to the right side of the screen, which will continually update as more information is added on the left. Then we need to load our target list, which is actually our mRNA cleaver output, which you can see down below. And then you'll import your results from intact mass. The program will automatically check off all of the digestion fragments that have the pass flag in the intact mass results table. What is considered a pass depends on your intact mass method. You can also manually check and uncheck fragments to include or exclude them from the coverage percentage and coverage visualization on the right-hand side of the screen. The coverage percentage is calculated here, and your visualization is here. You can capture image to clipboard and copy the coverage map into a program of your choice. And this was a quick overview of our sequence mapping data analysis workflow for nucleic acid digests. I hope that these overviews have demonstrated that intact mass, mRNA cleaver, and mRNA coverage mapper can help you effectively evaluate CQAs that are important to mRNA as well as other RNAs of interest.